Latijnse zitting werd gebed. Spiritus Sancti gratia illuminit sensus et corda nostra. Gaat u zitten. Welcome to this festive occasion, a festive day for the inaugural address of Professor Dr. Ole Jensen. Ole Jensen was born in Denmark, where he received a master's degree in electrical engineering from the Technical University of Denmark, after which he moved to the United States, where he did his PhD at the Brandeis University under the supervision of John Lisman on a thesis on oscillations, oscillatory short-term memory models. Then after a few postdocs, last one of which was in Helsinki, we attracted him to Nijmegen, to the Netherlands, in 2002, where he started his own PR group as the head of a research group in 2003 under the title Neural Oscillations Group. Ole has been extremely successful in the years that he joined the Donders Institute. He has published numerous publications in high-level journals, including Nature Review Neuroscience, and many more. He also has been cited almost 8,000 times, and he's been recognized by many others in the field as one of the leading figures in this domain of research. This was also recognized by the Society, so first in 2002 he received his Young Investigator Award of the Society for Biomagnetism, and in 2012 he received the Mid-Career Award. So we are extremely pleased that he has been able and accepted to be part of the college of professors of this university, and we're very much looking forward to his inaugural address. Professor Jensen, the floor is yours.
Yeah, thank you very much for this very kind introduction, and thanks to all of you for coming here today. So what I'm going to do is to talk about brain oscillations and how they help our basic brains to cope. So to set the stage for this, I will start by introducing by a book that was published now 45 years ago, almost, by a guy called Alvin Toffler, and the title of the book was Future Shock. So what this book was talking about is that society is changing faster and faster, amongst others due to new technology. So that means the technology is coming, or the future is coming to us at a faster and faster pace. So that would mean that some of us or institutions as such might be in a state of shock, in the sense that they cannot keep up with the rapid change and going on in society. Furthermore, he mentioned different things such like cognitive overstimulation would interfere with our ability to think, and that is of course relevant in the context of the ever-increasing information flow. He also pointed to an interesting notion that information overload, meaning our ability to process all the information in the context of our processing capabilities, if we have too much information overload, that can result in mental illness. Right, so this was 45 years ago. Obviously, the world didn't fall apart, right? Society is still working. I will now just show you an example of my own desk to illustrate how busy we all are, and then put that into the context of Alvin Toffler's book. So here you see a picture of my computer screen just in my office, right? First of all, up here to the upper left, you have emails. There are a few hundred of those clicking in every day. Then Skype, of course, is very important to be able to do video conferences with your colleagues. Furthermore, there's Spotify, because a bit of music at work is always nice. Then there's Facebook, such that I can keep up with family and friends. And furthermore, there's my iPhone, in case I have to attend a boring meeting. It's still nice just to be able to stay connected, right? So I should point to that this is by no means unusual. You go to any office and you will find that most people have these things going on the screen. Okay, that's only the technical part. Then during the day, Diego might be popping in, sharing some new ideas. Tilde might be calling, asking me to do the minutes for the next PI meeting. There might be Arne coming by in the window, very interested in showing his new data. And in the context of all this, what I really should be doing is to edit Jørgen's paper. Right? So nevertheless, I somehow managed to survive and get my work done despite all these disturbances. And I'm not going to make the claim that I'm exceptionally busy. I know we are all busy, have all busy lives. In fact, that has been documented in several publications, as you can see here. So then you might ask, so how do our brains handle all this information coming in? And really the stream of information has been increasingly rapidly over the years. It's probably going to increase even further, right? Furthermore, people are craving more and more information, as it seems, right? New gadgets, tablets, iPhones are hugely popular. But how do our brains happen to manage and prioritize all this information? Well, you might say that's easy. Our brains have 100 billion neurons, and each neuron has 10,000 synapses, so all in all there's more than 1 quadrillion connections. So in short, the brain has a huge processing capability. However, that notion sort of conflicts with our common sense. Just assume that you're driving on a highway and you're passing this guy, right? A guy driving fast, talking on his cell phone. It makes you a little nervous. Why is that? Well, that is because we all know that we have limited processing capabilities. And cognitive neuroscience has many examples of this, right? For instance, when it comes to working memory, you can only keep maybe 6, 7 items in your working memory. When you're talking with other people, you can only attend to one person, and so forth. So really, we cannot do that many things at the same time. Our processing capabilities are limited. And to explain this, I'll just give you an example from the visual system, where we can point to some very concrete bottlenecks in the visual system. So here you see an example of G. Ole Bergman. So just imagine I'm sitting in my office having a meeting with Till. So of course I can recognize Till no matter where he is in my visual scene, and indeed Till's face is represented in my early visual cortex. However, you also know that if you go down the visual stream, there is an area responding in particular to faces, the face area. And these neurons in the face area are becoming active no matter where Till's face is. So far, so good. Now say that Mathilde is joining the meeting. So now I have 2 faces in front of me. As you now can see, that might pose a problem in this hierarchical system of the visual system. And one could believe that these 2 faces might merge together. And then something rather disturbing would happen. As a fact, this does not happen. So what that means is our visual system does have the ability to keep multiple, say, items or representations apart. So how does the brain do that? We have now conducted a lot of research, and that has led us to believe that oscillatory activity, in particular oscillatory activity in the alpha band, is very important for doing this. And to explain this, I will show you an example of a typical EEG recording. So this is an EEG recording of Ali Matahedi. So basically, what we measure is the electrical field on the scalp with a simple amplifier. The first recordings were done in the late 20s by Hans Berger, and the typical observation is that when people sit there and are relaxed, you see ongoing oscillations at 10 Hertz, meaning 10 cycles per second. And that is the alpha band activity. However, when people then get excited, the observation typically is that these alpha oscillations are diminished, they are reduced. So this has led to the connotation that alpha band oscillations are associated with a state of being relaxed, or also called an idling rhythm. What we have found evidence for is that that notion of alpha activity reflecting rest is simply wrong. And to us this is very important, because really the alpha oscillations are the strongest signal that you measure from the human brain in EEG recordings. And we really believe now that these alpha oscillations have a very strong functional role, doing much more than just reflecting rest. And I'm now going to explain what our thoughts are on this. And I'll do that by pointing to a working memory study we did now almost 15 years ago. And first I will introduce the notion of working memory. What is that? Well, it's very simple. So say here that you have two people. It might be here an example from the movie Casablanca, the uncut version. Here you have Ilsa, and she's telling her phone number to Laszlo. She wants him to call in. Laszlo then has to remember the phone number. In other words, he has to keep it in the working memory such that he can write it down, right? So this is simply what we mean by working memory. You have to keep some information online. So what we did some years ago was to get our hands on some EEG data. And we were very interested in oscillatory activity, in particular in the theta and the gamma band. And we operationalized this working memory task such that a set of digits, in this case 36501, were presented to the subject. Then there was a retention period of a few seconds, and then a probe came on. And then the subject simply has to say yes or no to whether that probe item was on the list. And what we furthermore did was to vary the number of items on the list to 2, 4, or 6. And then we used EEG to ask what was the brain state when people were maintaining these working memory items. And when we did this, first of all, we got a surprise. So what you see here to the right is a power spectrum, and it signifies how much oscillatory activity there is during this working memory retention. And you can see there's a strong peak at 10 Hertz, right? So this 10 Hertz peak means that there is alpha activity during working memory maintenance. However, the real big surprise came when we compared the number of items to the alpha band activity. And what we then observed was a systematic increase in alpha band power with the number of items you keep in working memory, right? So this here can by no means that alpha reflects a state of rest or idling, right? Then alpha will go down. We see alpha activity go up the more difficult the task is. So what is going on here? Well, basically what we back then proposed and still believe is that this alpha activity in the back of the head reflects an inhibition of the visual system, right? So you have probably observed, or you might do it yourself, if you really have to focus, you sort of stare blank up in the air, right? You probably do this to prevent interference. Now it turns out that it seems like the brain has mechanisms to do exactly the same, and that is the alpha band activity. It simply serves to shut down the visual system to keep up interference, to keep out interference. Now we have taken this notion and then explored that in different kinds of studies, namely the inhibition by the alpha activity. And what we then have, what that has led us to propose is a more general theory in the sense that we talk about gating by inhibition. So if you now consider the brain and a subject doing a given task, then of course there's a lot of networks in this brain, and these networks are connected. Now that subject has to do a task, and we know that that task then requires a certain number of networks to be active and also to communicate. So then you might ask, how is that communication or this functional connectivity between these nodes in the network established? We know that it cannot be done by new connections forming and so forth, because that's way too slow. There has to be something about the dynamics of the brain activity. And what we are proposing is that this is achieved by simply shutting down all the regions not needed for the task of hand. that then leaves the regions that should process the task to communicate. Furthermore, we also believe that alpha activity is reflecting the shutting down of the task irrelevant regions. So this is a notion we have been tested over the years, and a very complete hypothesis of this notion is that in order for you to perform optimally in a given task, there need to be a shutdown of the relevant regions, and this shutdown of the relevant regions, the inhibition, as reflected in the alpha band activity, should correlate with how well you perform, right? So what we have been looking for is tasks where you actually perform better if alpha activity becomes stronger. And I'm just going to show you a few examples of these type of tasks. So let's go back to the scene here with Ilse and Laszlo, right? So Laszlo is there, he has to write down the phone number. In comes Rick, he's the ex-boyfriend, and he's a bit on the jealous side. He wants to prevent Laszlo from remembering the number so he can write it down. So what Rick does is simply to say a random string of numbers. So we can also now see if Rick managed to suppress this interference, he'll be able to write down the phone number and he can later call Ilse. But if not, he will miss out on that opportunity. Okay, so this type of setting we have then operationalized into an experiment. And this is an experiment that Mathilde Bonfant has been doing. And basically, you go back to the good old working memory task, you present a set of digits and a probe comes up. What Mathilde then did was to introduce a distractor, right? So that distractor could either be another digit, that would be Rick, just saying random numbers, or it could be a weak distractor, just as a symbol, right? So very important here, subjects could anticipate whether a weak or a strong distractor was being introduced. And then what we simply did was to ask, what was the brain state in the anticipation of an upcoming distractor? And in this case, we used MEG. So MEG is sort of the bread and butter tool for my group. And with MEG, we have 275 sensors distributed over the head. We measure signals very similar to the EEG, namely the oscillations, but what MEG also allows us to do is to identify where in the brain the activity is being produced. So Mathilde conducted this task, and the findings came out very clear. So what you see here is a color plot. This is a time frequency representation of power. And the stronger the color, the stronger the oscillations. And what you should note here is that just before the distractor, there's alpha activity turning on, right? Furthermore, Mathilde also compared the strong to the weak distractors. What she then found was that there was more alpha activity for the strong as compared to the weak distractors, right? So this is really a nail in the coffin to this resting state notion of the alpha activity, in the sense that alpha activity is shooting up, and it seems to serve to prevent the shutting out of the distractors. Furthermore, since we're using MEG, we could localize this activity to early visual regions. So then you might ask, does this alpha activity then also help you to perform the task better? And one way to go about that is simply to look at how fast people respond to the probe. If they can respond fast to the probe, they are probably doing better as compared to when they respond slower to the probe. So what we then did was to sort the trials into fast and slow responses. And when we did that you can see at the gray bar, what we observed is that the alpha activity was stronger for the fast trials. So in other words, if you do not manage to increase your alpha activity just prior to the distractor, you will be punished in a sense that you are doing worse on this working memory task, right? So this is to us really strong evidence for the alpha activity being functional, and if it doesn't turn on in regions that you don't need for the task, you cannot perform optimally. Okay, so far we have just been talking about alpha activity recorded from the scalp, right? But of course, you all know that it's neurons inside the brain that is doing the job. So then you might ask, what are the neurons doing? How are they either contributing to the alpha activity, or how are they being modulated by the alpha activity? So this is, of course, very difficult to study in humans because we very rarely have access to individual neurons. However, we were so lucky to establish a collaboration with Ranulfo Romo in Mexico. And his group has been performing multi-site recordings from monkeys, and that allows us to look at both individual neurons and also population activity. So it was Saskia Higgins in my group who established this collaboration, and then she went to Mexico to look at Romo's data. Because what Romo has been doing so far was to look at only the spiking activity. And this is what you see here in the lower trace. That is recording from individual neurons that are spiking. And according to him, he was somewhat sympathetic to oscillations, but he said, we just never see these oscillations in our spiking data. Nonetheless, what they have never come around to was to analyze the so-called local field potential data. And the local field potential data reflects the population activity of a greater set of neurons. And it's also the local field potential that we think is related to the MEG activity we measure outside the scalp. So when Saskia then did the analysis of the local field potential data, we got quite a positive surprise. So what I'm showing you here is a case study, data from one monkey. And what we see in that monkey is that indeed there's strong alpha activity in sensory motor regions, no matter what region we looked at. So in somewhat contradiction to Romo's notion, there is actually oscillations in these individual regions. So what might be going on? Well, what Saskia did next was to go in and look at this alpha activity, and she simply cut out snippets of data. Then for each snippet she calculated the alpha power and then sorted the trials. And this is what you see on the x-axis. What she also did was for each snippet to go in and count the number of spikes per time. That allowed her to correlate the alpha activity with the number of spikes. And what she observed was a systematic decrease in spiking activity with alpha power, right? So this is actually somewhat a big surprise in the sense that the strongest activity that we measure outside the head is actually associated with a decrease in firing. What Saskia also observed was that it's possible to look at the individual spiking and relate that to the local field potential. So it's called spike field coherence. So what you should note here on the lower part of the plot, red means the stronger the color, the stronger the spike field coherence. So what this here means is that when you have spiking, then this spiking is clogged by the ongoing oscillations in the local field potential. So what might be going on here, right? So in order to sort of illustrate what's going on, I'm going to do a little experiment. And I'm going to do that just with you guys sitting over here, right? So when I lower my hand, you have to start just clapping ahead. That's all there is to it. Start. Okay, thanks, enough. Now comes experiment 2. Now you just have to clap very hard to congratulate yourself for doing so well. Thanks. So, what's going on here? Well, first of all, you noticed that the people started to clap rhythmically very fast, right? So there was somewhat an emergent property, meaning that people are somewhat listening to each other and then very fast get into the same pace. There was no one conducting the audience here to go at a certain rhythm or synchronization, right? It just emerged spontaneously. So that's observation 1. The second observation is that when the audience was clapping very fast or rapidly, then of course the firing rate or clapping rate, if you will, was faster when you saw the rhythmic clapping. Now it turns out that the signal that we measure from the brain corresponds to the sound wave of all people clapping together, right? So individual people clapping you can consider as neurons. Of course, neurons also communicate, and it turns out that they also lock up in synchronization very fast. So the key point here being that when we actually observe the rhythm in the population activity, meaning the whole audience clapping, you actually have fewer claps per second. So this is exactly the same we think is going on when we think about the alpha activity. We think that the alpha activity is a consequence of pulse inhibition, right? So these pulses of the alpha activity is preventing the firing in a rhythmic manner. However, when the inhibition dies out, the neurons, they can fire strongly, right? So this scheme here exactly explains why there is a decrease in the firing rate as the alpha power is going on. And this will also support the notion that the alpha activity is doing something like functional inhibition by shutting down neuronal firing, or at least reducing neuronal firing in a phasic way. Okay, let's now go back to the notion of the future shock book. It was also put forward that mental disorders could be associated with an inability to handle all the information coming into us. And certainly if you look at the statistics, there has been an increase in attention deficit disorders over the last decade or so. So as we now put forward the notion that the alpha activity has something to do with the inhibition of these distractors, and we know that patients with ADHD are quite easily distractible, we put forward the hypothesis that it could be that this distractibility is associated with an inability to modulate the alpha activity. So together with Niels Bühne, PhD student, and Jan Beuteler, professor in psychiatry, we conducted an experiment on ADHD. I will first try to explain the paradigm we used. And again, we are starting with the notion that alpha activity is gating by inhibition. So we have developed a set of experiments to further explore that in a more spatial attention kind of setup. So what is spatial attention? So imagine you are running around in a jungle. Suddenly there's a tiger showing up to the left, right? Of course you need to do something about that. However, it could also be that there is a distractor coming up on the other side. It could be Angelina Jolie. To some of you, Brad Pitt might be more distracting. But there's a distractor coming up on one side, but in order to increase your survival chances, you better suppress that distractor such that you can process the tiger. We also know now that what is presented in your left hemisphere is being processed by your right brain hemisphere and vice versa. So there's literally this crossover. And when you do these kind of tasks, what we see is that when you are attending to the left, the alpha activity is going down on your right hemisphere, whereas the alpha activity is going up over your left hemisphere. And we really think that this alpha activity going up over the left hemisphere reflects the ability to suppress distraction. And this is what we think might be impaired in ADHD patients. So to study this, Niels designed an MAD experiment based on spatial attention. So what subjects got here was a simple cue. It told them to attend to the left in this case, and then a few seconds later a target came on that was difficult to detect, and the subject has to say yes or no to whether this target was there, right? So now we can measure people's ability to make use of this cueing information, in the sense that if we do not present a cue, people would be a bit worse, but if we present a cue, people would be a bit better. Now it turns out that ADHD patients actually do not benefit from this cueing in general. And we can develop a measure for how well they benefit from this cueing. And now it turns out that the more ADHD they have, as in this case measured by a self-report, the less they benefit from this cueing. So you can also argue that they're sort of open to everything what happens out there in the visual scene, or you can argue that they do not benefit from the spatial cueing. Both is correct, whether it's good or bad is of course a matter of debate, because it depends on the context we're doing. Nevertheless, when we do this kind of experiment with MEG, what we see is a very typical thing. and that is that when people are attending to the left, their right hemisphere is being suppressed in terms of the alpha activity, or the alpha activity is going down, and that is reflect increased processing capabilities in your right hemisphere. However, the alpha activity is going up over the left hemisphere, as you see here by these time frequency representations. What we now can do is to look at the 10 Hz activity only. And when we do this and compare controls, normal healthy adults in this case, to young adults with ADHD, what we see is that the alpha activity breaks down at the end of this interval, right? So the ADHD patients can simply not maintain their attention as reflected in the alpha band activity. So what is going on here? What is going wrong in these patients? So you might suggest that it's an inability to generate alpha activity or sustain alpha activity in the visual system, or it could also be due to an inability to exercise the top-down control, right? So it could be that it's the frontal regions that are not able to exercise the top-down control on the alpha activity as well as one would like to. And it's actually the latter that we think is the case. So we have not been able to investigate that systematically in ADHD patients, because what we need to do is to uncover the full frontal network doing this top-down control. But how do we do this? Well, there are several candidates for doing the top-down control. Wouldn't it be amazing if we actually could go in and sort of in a reversible way lesion a frontal region and then see what the consequences are for the posterior alpha activity? If we could do this, then we can also try to unravel the networks being involved in doing the top-down control. It turns out we can do this. Basically, what we do is that we hit the brain with a very strong magnetic field. It's called transcranial magnetic stimulation. So we impose that very strong field, in this case, for instance, on the frontal eye field, and that might then impair the top-down control and thus the ability to modulate the posterior alpha activity. So I'll show you how we go about this. So here you see Tom Marshall doing a TMS experiment on Mathilde Bonfond. And what Tom has in his hand is a coil. And this coil is just a figure 8 wire that we send a lot of current through that generates a strong magnetic field. You repeat this in bursts, and that lesions over a short time a given region that we can then identify in the structural MRI. But let me put it the other way around. We have the structural MRI of the subjects that is on the screen you see to the right, and then Tom can guide the coil around in order to identify the region he wants to lesion. So what he has then been doing is to conduct this type of experiment, do the lesioning, and then very fast reel the subjects to the MEG system, and then subject the subject to one of our cognitive tasks, again a spatial attention task. And the task he used was quite related to the one we saw for the ADHD patients. Basically, there's a cue telling the subjects to attend to the left and right, and there's a target coming on. Again, when we did this experiment, what we see is this very classical alpha lateralization, right? You attend to the left, alpha goes down in the right hemisphere, it goes up in your left hemisphere in order to suppress potential distractors. Here then comes the results when we perturb, in this case, the frontal eye field. So I should just tell you the frontal eye field is one of the frontal control structures that we think are involved in doing the top-down control. What you see on the lower figures is what happens when we do the FEF stimulation. And what you should know is that the alpha activity is being abolished, or the modulation of the alpha activity. However, if we do the control stimulation on the center of the brain, the alpha modulation is sustained. So this here tells us that the frontal eye field is causally involved in doing the top-down control. This is, of course, just one region implicated in this top-down control, and we need to work a lot harder for many more years in order to recover the full network. However, when we have recovered the full network, then we can also take this research to, for instance, ADHD, to ask more mechanistically what is going on in these patients when it comes to attention or control. So this was some examples of the experiments we have been making over the years and how we are building our case. So I'm now going to switch gear and tell you about some of the future directions. So as you probably remember from the clapping experiment, when audience were clapping at the rhythm, of course the timing of the clapping is highly controlled by the population activity, right? So in other words, there is a phasic clocking of the clapping. What we would like to know more about is how is this phasic control involved in controlling when neurons that fire or not, and how might that benefit computation in general for what's going on in the different brain structures. So I'll outline some of the ideas we have on this. So let's go back to a complex visual scene. So imagine that you're on a bicycle, you are going to work in the morning. There's a lot of traffic, a lot of bikes around, of course. So of course it's very, and all this information, when you're doing this task of biking to work, of course there's a lot of information coming in parallel, right? All the visual information from the other bikes, the cars, the pedestrians, the street lights and so forth, right? And all this has to be processed, but the point being that it's massive, the amount of information coming in, right? So in this case, of course, you need to attend to the lady on the orange bike in order not to ram into her, right? So then you might ask, how does the brain solve this task? Well, many have proposed that spatial attention is very involved in this, and that is of course correct. Like you sort of zoom in to the lady on the orange bike and then process what's going on in that part of the visual scene. However, that is not the full story, right? In the sense that you also need to be ready to process the guy on a yellow bike. You might even also attend to the traffic light. So there's all these things in the visual scene that you need to prioritize. And the classic notion of spatial attention is not sufficient to account for that. So the idea we are trying to develop is that the brain is making small to-do lists in order to prioritize a visual scene, or is breaking this visual scene down into a sequential code. And what we think is going on is, as I mentioned before, alpha activity is oscillatory, is also inhibitory. So that means that you have inhibition going up and down. When you are in the peak of the inhibition, neurons in the visual system will not fire. But as the inhibition decays, only the most excitable neurons will fire, right? In this case, it might be the lady on the orange bike. A little later, you go down the inhibition, the next item of representation will fire. That would be the guy on the yellow bike. And then after that, it might be the street light, and so forth. So through this process, we believe that the brain is making these more to-do lists or sequences of visual representations that then can be processed. And if this is true, it would be quite amazing, because it also implies that there's a phase coding going on in the visual system pertaining to the alpha oscillations. And I should say that this is only hypothetical so far. A lot more work has to be done to either falsify or support this notion. And also another interesting question that we have some answers to now is, how is the excitability of these individual representations being set? Because of course it's the excitability determining the order of activation. I'm now going to switch to an even broader note, and that is to talk a bit about big data. So you have probably all heard about big data out there. Due to computers being more and more powerful, digital storage devices being bigger and bigger, it's possible to collect big data in all fields, right? Economy, neuroscience, what have you. Now it turns out that in cognitive neuroscience and brain imaging, we also has the ability to collect a lot of big data. Just as the Douglas Center, we have 3 MR systems and an MEG system. And of course, just during a day, we can crank in gigabytes of data, right? Or a year, we can crank in terabytes of data. Of course, we also have the ability to collaborate with other centers and make really huge studies, and then we can collect petabytes of data. So these data, of course, allows us to look into the brain. So maybe by having all these data, we will finally be able to understand how the brain works. No, I don't think that is going to be the case. I'm not going to argue against big data collection here, but I think when we also collect these big data sets, we also have to interpret the data in terms of big theories. Of course, otherwise the whole endeavor doesn't make sense. Don't get me wrong, I'm not against collecting big data sets and having multi-center collaborations and so forth, but I think these data collection endeavors should be done in the context of theoretical frameworks for brain computation. I'm not now going to claim that I have these theories that should guide the data collection. However, I would like to point you to some of the lessons we have learned that we think to point to sort of a more general framework for how the brain operates as a network. And the first idea I introduced to you was the notion of routing by inhibition. So again, this pertains to, we have a number of brain regions, they have to communicate in a given task. What we have been proposing is that this communication is set up by shutting down all the task-irrelevant regions. I've given examples for how we can study this notion by looking at brain oscillations. However, of course, you can also study this framework just by doing functional MRI, and then you can look at the board signal, and then you can ask, is the decrease in the board signal in certain regions predicting of performance, since it might be reflecting inhibition of these regions. And indeed, there are studies out there suggesting that. But what I propose is that maybe that could also be put into a more general framework in which routing is achieved by the inhibition of the task-irrelevant regions. The other idea, so we have been putting forward, is the notion of parallel to serial conversion. So again, we have the visual scenes, but we also have the other sensory modalities. The brain is bombarded with a lot of information. This information, in order to be handled by the brain, has to be broken down to serial codes or serial lists that are prioritized according to importance. And I gave you the example of how a visual scene could be broken down by oscillatory activity, inhibitory oscillatory activity, that then allows the sort of release from inhibition and the sequential representation of the, say, most pertinent visual representations. The notion for parallel to serial conversion might also apply to the memory system. Here's an example of Oscar, my son. He is sitting there thinking about what he wants for his birthday. And when thinking about, there's a set of memories popping up, right? We also know that these memories are encoded by synapses in recurrent networks, or at least that's a very strong theory for how memories are encoded. So what we now also think is that these memories are sort of competing to activate, and the activation is determined by the recurrent excitation defined by the synapses. Also in the memory system, the hippocampus, you have inhibitory theta oscillations, and this inhibition dies down during the cycle. We then think you can get memory recall by allowing the most excitable representations to activate sequentially. While this might sound somewhat speculative, actually if you look at the research literature on phase precision and so forth, there's actually some indications that this notion could be correct. But in other words, the bigger perspective here is that these oscillations allows for a parallel to serial conversion. So I'm going to end here. I'm going to summarize. I've put forward the notion that information is routed in the brain, and we think that alpha inhibition is strongly involved in doing this routing, namely by keeping the task-irrelevant regions inhibited. Of course, in order to understand this picture full out, we need to understand how these alpha oscillations then are modulated, probably by frontal regions. We need to uncover that. And that also has the potential of allowing us to look into different kind of disorders associated with attention deficits. Furthermore, we have been putting forward the notion of parallel to serial conversion by oscillations. That is something we are going to explore much more in the future. But the basic idea is that these oscillations allows for the creation of small to-do lists that are prioritized according to importance, and these small to-do lists then prevents the information overload in the brain, in the sense that they allow you to focus on only the most relevant things to be processed. So I'm going to finish here by acknowledging people being involved in these studies. First of all, I should acknowledge Peter Haggard, the gentleman to the left. So more than 12 years ago, now Peter Haggard took the initiative to start the Dundas Center. Not only did he make sure that we had all the right equipment, brain imaging equipment, but he also made sure to have all the right people to do the research. And as a consequence, he has created a very vibrant and intellectually rich research environment that we all have flourished in. Now I think hundreds of researchers, due to Peter's efforts, have now been able to have very successful careers in cognitive neuroscience. And of course, all the research in the Dundas Institute would not be possible without the support from Tilde, Eric and Arthur and their respective teams, who have been very devoted to keep the infrastructure of the center going. Furthermore, I should also acknowledge John Lisman, is my old PhD advisor. He was the one who put me on track to look at brain oscillations. I'm still working very much with John, and we are still meeting regularly to exchange ideas. But also very importantly, I should acknowledge all the students and postdocs and collaborators who actually have been doing all the work over the years. I've been very privileged to work with a great number of very talented, young and enthusiastic people that have made all the research possible. I should also acknowledge my family and friends coming from far away to be here today. It's very special to me that you have gone through the efforts to make it all the way to Nijmegen. And finally, I should acknowledge and thank Freya and the boys, Oscar and Felix, for always be there with providing so much and joy and love. Thank you very much.
Dear Professor Jensen, thanks a lot for this wonderful overview over a scientific adventure. A scientific adventure in which you and your group have almost single-handedly changed a dominating view on the role of alpha as a brain signature on cortical iron into a much more interesting and also now widely accepted view that alpha is actually an active mechanism for inhibiting task-irrelevant areas. And that overview gave us wonderful insights in the work. But you've shown also in this lecture that you're not only a great scientist, you're also a gifted and great teacher. Actually, Dan Wagner, a famous cognitive scientist, a number of years ago gave his subjects the instruction, don't think about the white elephant. And it turns out to be the case, it's very difficult not to think about the white elephant. Now, all of us have our own white elephants, but you managed in your lecture to induce enough alpha activity such that we were able to suppress thinking about the white elephant and focus on the content of your talk. So the gift of teaching is to establish a synchronization between the brain of the teacher and the brain of the audience, and that's what you have shown that you're able to do. So this university not only should congratulate itself having you on board as a scientist, but also as a teacher, and especially the Faculty of Science that appointed you as their chair in neural oscillations. I would like to congratulate with your appointment as professor in neural oscillations. So also, of course, congratulations to Freya and the family. and to the whole academic community having you here on board. Now, before I close this academic session, a few remarks on the organizational announcements. Before you all leave, please give the cortege the possibility to leave the room after the Ole Jensen and his wife, and also the partners of the academic colleagues are allowed to join the cortege. Now, once downstairs, where in the Anton van Duinkerk hall there will be the recipe to congratulate Ole Jensen with his inaugural address. Don't stand in line, but just take a drink, because that's been supported for this kind of occasion. And wait until the moment is there in order to shake hands, because otherwise you have to wait for a long time, and that might not be the most optimal way of celebrating this inaugural address. So please take the opportunity to get a glass and wait for an opportunity to congratulate Ole for this occasion. Okay, then I'm going to close this academic session, and I ask you to raise. Just die Animus omnipotens.